Okay, the recording started. Okay, so we are, as hard as this is going to be to believe for you, we are just about a month off of the end of the semester. Without spring break, I know, right? Everybody, go ahead and hit your little clap button. Where's the clap button? I'll hit the clap. There's somewhere here. I'm in. How come I don't have clapping? There we go. Clap it. This has been brutal for everybody, I think, and I just I hope you're getting what you need out of the course. I think mostly the genetics courses and some of the lectures are okay. I do regret that we're not in the lab. I mean, I think that's the biggest deficit, where you don't get an opportunity to get some hands-on experience. Analyzing data is great. I think the experiences that we've tried to put together for everybody using the computer are really good, because so much of biology these days, and especially genetics, is done on the computer. I should point out that this week are the Drosophila genetics meetings, and they are all done online and on Zoom. So another part of my life where uh, there are no people involved, which is really tough. These meetings are usually great places to connect with colleagues. But I will say that there is just a huge amount of work that's being done with already existing online databases, spreadsheets, programming, you pretty much name it. So I will say this again in a couple of weeks, but I highly encourage you guys, if you have the opportunity, to take either certainly bioinformatics. Dr. Luke and I usually swap years teaching it, so we alternate years. And I think Dr. Seiler is going to get involved as well. So we've got some really good people, and I'll excuse myself from that. Doctors Luke and Dr. Seiler are fantastic, and it'll give you an opportunity working with some of these databases, and really doing some of the things that we're going to do today, and certainly things that encompass the remainder of the course. And this is really where we're headed. So I highly encourage that. Also, I teach systems biology usually every other year. Um, and we talk a lot about programming, how to probe databases, how to use software to create unique graphical figures to explain very complex topics. So this is really the direction that a lot of biology, and very, very specifically genetics, is taking. So we're going to talk a little bit more about these things this week. Just to give you a heads up, I think we're due for another exam. You should be finishing this week all of our mechanisms of central dogma, obviously replication, transcription, translation, and everything up to translation, and some of the things that we talk about here will be on the exam. And I think next Friday is as good a day as any to have the exam. The format's going to be a little bit different than the previous exam. I'm not entirely sure. Um, I'm not entirely sure because I haven't written it yet. And as soon as I do, probably uh, next over the weekend, I'll take a first stab at making the exam. I'll let you have more details about it. But it's really about the mechanisms. I mean, that's really where we are. And some of the things we'll talk about today, especially, these will end up on the exam. These problems will end up on the exam as well. So let's go ahead and take a peek. I'm going to share my slide screen here. And oh, bad news. Hold on 1 second. Somewhere I thought I was going to pause the recording, but I guess not. Don't panic. I forgot to plug in. There we go. If you hear a beep, it's my little pen. There we go. Sorry about that. I knew I forgot to do something, but I got involved in reading a new paper that was published in Science magazine about antibody titers for SARS-CoV-2, which is, of course, SARS coronavirus 2 is the virus that causes the disease COVID, and really interesting results on people who've been vaccinated with the mRNA-based vaccines, really showing the increase in antibody titers in response to the mRNA vaccines. So great stuff. If you get a chance, read always be reading Science magazine. Now I am going to make sure things are working here. And indeed they are. Cool. And let's make sure I get the right color. I don't know why red always shows. Is it red? Yep, good. So basically, I want to review something that tends to be a little bit complicated, especially for new genetics students, and I want to be in a little bit more detail. Here is a simple single strand of DNA molecule. And by the way, all these problems, etc., are in the problem sets folder on Blackboard, so you can get a hold of them if you would like them in a different format than on this lecture. And if you're given a single strand of DNA, what you're very often asked to do is, what is the complementary DNA chain? Please don't spell complementary with an I. Okay, if you have a complementary DNA chain, that would be something that says, oh my, what a nice DNA chain you are. You're giving them a compliment. Okay, these are complements. Okay, the vowel here makes a big difference. So when we're talking about our complementary DNA chain, so what are we going to do? We're going to create the DNA chain. What would it be? So how would you start? I'll just write it underneath here. So what's the first thing you got to realize?
Change it to 5' to 3.
You got it. Excellent. Always, always, always annotate the 5' and 3' ends of either the DNA and or the RNA that you are working with. Direction matters. Okay. Anti-parallel strands are anti-parallel for a reason. Writing the 5' or 3' end of the molecule certainly helps to remind us that the DNA strands are anti-parallel, and it also orients you on the molecule themselves. So it's really important to get that straight. As you progress, meaning if you get out of undergraduate, go to graduate school or some professional school, very often the convention is to always write DNA or RNA sequences in the 5' to 3' direction. And that's that's just the way that everybody does it. However, it is very important at this point in your genetic education, it is very important to write down the direction. And the reason is clear, is that you have to get used to the whole idea of anti-parallelism and complementarity, and get that straight in your mind before you get used to using an abbreviation. Okay, so this is the really important part. So if we're going to make, we're going to go from 5' to 3' end, and it's fairly simple. So what would be the complementary DNA chain? Well, T binds to what?
A.
A. Oh, yeah, piece of cake, right? T. I'm assuming all of you, that's a G in case you were curious. Right? Is that correct?
No.
No.
No. How is that possible?
You put a C matching with a T instead of an A.
Oh, gosh, you're right. Don't believe me. You guys know the rules, okay? And we all make mistakes at times. So, yes, you should pay attention and watch your sequences all the time. So now the question is, what mRNA chain, and again, we're only working with small sequences, that would be transcribed from this particular strand of DNA? And this part of the question is rather confusing, and in the sense that you have to remind yourself of the way transcription works. And I absolutely love this little silly diagram. It is very simplistic, but in the same token, I think it communicates very nicely all of the things that you need to know about transcription in summary format, okay? So in order for transcription to occur, notice the orientation of the 5' to 3' and 3' to 5' strands of DNA. 5' is oriented to the top, again, a little different than we had up here, and intentionally so. The convention is to put the 5' to 3' strand on the top and the 3' to 5' on the bottom. Now, notice if we were talking about replication and we were talking about the primary enzyme involved in replication, DNA polymerase, what direction does DNA polymerase move on the DNA strand? Okay, you guys remember? DNA polymerase always moves.
Doesn't it move? Doesn't it move from the 5' to 3'?
Yes, indeed. Good job, Ash. It always moves in a 5' to 3' direction. Always, always, always. You'll notice RNA polymerase down here at the bottom always moves, in a 3' to 5' direction. So this is the big difference between replication and transcription. Replication and transcription. So what's required for transcription to occur? We must have, of course, here is our promoter region. The promoter region very often in terms of molecular biological shorthand, this is the transcription start site. The transcription start site, TSS. There are several proteins involved that bind to the DNA, not just proteins, by the way, but certain bits of RNA bind to the DNA here. And what they provide is a landing, basically what I like to think of as a landing strip to tell RNA polymerase that, okay, this is where you bind. This is the bit of DNA that we are going to transcribe. So there's a little bit of space in here where our RNA polymerase lands and then begins to move forward on the DNA. Now notice that there are a couple of base pairs here, and there are several. It's just illustrated here to make the cartoon look nice as just two. They're in gray. Okay, they're in gray. So what we're really doing, so it begins here. So we've got the transcription start site. RNA polymerase is attracted to the proteins and the RNA here. It begins to slide on down to the double-stranded DNA and begins the process of transcription. RNA polymerase, moving in the 3' to 5' direction, creates a message, mRNA, indicated here in bright blue, because it's reading the strand in the 3' to 5' direction, it creates the complement in 5' to 3' direction. And as such, we always call the 5' to 3' strand of DNA the coding strand. Remember, this is the DNA. Sorry, that's an N. My pen's not, must be out of ink here. That was a joke. And the 3' to 5' strand of DNA is called the template. It is called the template because the RNA reads the template and creates the message from the template. But because of the binding of the nucleotides, it turns out that this sequence of messenger RNA, and we could use our 5' to 3' right here. So if we were to write this up, we would know that our 5' to 3' is A, T, G, G, C, T, and I may not get these all right, C, A, T, G. Okay, A, T, G, G, C, T, C, A, T, G. All right, which means that the coding strand will basically match the sequence of the messenger RNA, both in a 5' to 3' direction, with one minor difference. What's the minor difference between the sequence of the coding strand and the sequence of the messenger RNA?
RNA has uracil.
Absolutely. So that means that when we look at our messenger RNA, we'll end up with A, U, G, G, C, T, C, A, T, G. Oops, what'd I do? All of our T's
All T's are used.
to be uracils, right? Okay, so this prevents a little bit of a conundrum for students. and I include myself in this bucket of students, okay? If you want to know from a biochemical perspective how RNA polymerase works, and you want to know the sequence of the messenger RNA, basically you can act as if you were RNA polymerase riding in the 3' to 5' direction up here and just begin to create the complement. So if I know it's T, it's an A. If it's an A, it should be a T, but we know it's messenger RNA, so we know it's got to be a U, G, G, C. So you can behave as if you were RNA polymerase and go along the template strand in the 3' to 5' direction and create your DNA. For some people, that process is a little bit difficult because you're doing multiple things at once. You're taking into consideration the anti-parallelism of the DNA strand. You are taking into account the complementarity of the nucleotides, and you're also replacing all your thymines with uracil. So a much simpler way that many students find is to just take the sequence of the coding strand and just replace all the T's with U's. It is entirely up to you, as long as you understand both methods, it's entirely up to you how you want to convert the sequences. It's entirely up to you. Now, something that we do know, and this is really something that gets highly complex when looking at genomes, is our little cartoon here implies that one of the strands is always the template strand. So one of the two anti-parallel strands in DNA is always the template, and the other is always the coding. And this could not be further from the truth. I mean, if you think about a linear bit of DNA, okay, which one's the top and which one's the bottom? And the answer is, all right, I can't read my own writing, sorry. If that's 5' to 3', then this one is 3' to 5'. And okay, so which is the top and which is the bottom? Does the orientation matter? And the answer is no, it doesn't. So this means that for any stretch of DNA within a chromosome, one strand or the other could serve as a coding or template strand. It just depends upon the gene in question. So try not to get yourself too oriented to thinking that only one strand of DNA could possibly be coding and one strand can be template. And in fact, there are many genes that behave where the complement. So for this might be gene 1. And in gene 1, the 5' to 3' in this direction is indeed the coding strand. But for gene 2, the alternative strand could be coding. And there are many genes that overlap in the two directions. So the organization of the genome is much more complex than our little cartoons seem to imply. So cartoons are great. Remember, things are much more complicated. Now, there's a reason I chose this particular sequence. Anybody remember from their reading why this sequence that I chose was important? And you'll get a clue from down here. This is one of those things that your mind, as you continue in genetics and molecular biology, your brain will immediately recognize. And that is, in addition to our transcription start site, our RNA polymerase coding and template strand, in order for RNA polymerase to begin the process of transcription, not only do we need a transcription start site, but we need a start codon. And in DNA lingo, our start codon is ATG, always 5' to 3'. And of course, the related messenger RNA is AUG, 5' to 3'. So transcription start site attracts the RNA polymerase. RNA polymerase hits the landing strip. It starts moving down the DNA strand until it hits a start codon. Once it hits the start codon, bam, it begins creating the messenger RNA. And it putters along until somewhere down here, it hits a stop codon. It hits a stop codon, and then will progress for a period of time until it hits a transcription termination site. And then the RNA polymerase releases from the DNA strand. Okay, so we've got transcription start codon, stop codon, transcription termination site. Each of these positions has a characteristic DNA sequence. That's how everything knows how to interact with DNA. It is a code. It is a characteristic sequence. These sequences, and there are more than one, at least for transcription start sites and termination sites, are how computers count genes. If we have the complete genomic sequence of all DNA in any genome, we can count the number of genes by identifying transcription start, any sequence of gene that has a transcription start, stop, transcription terminate. So we look for those 4 elements. And we use computers to do that, which is why, of course, you need to take bioinformatics. We use computers to do that, and then we calculate the number of times those 4 elements occur in a row, and we can count up genes. And that's basically how we know there are 22,000 some odd genes in the human genome. We don't actually know each and every coding sequence, each and every bit and detail and protein sequence, but we do know that they have those 4 elements in a row. So when we say there are so many genes in the human genome, what we're really saying is we know that there are so many pieces or strands of DNA that have transcription start codon, stop codon, transcription terminate. So if you think about it, again, our purpose here is to look at all the multiple definitions of a gene. We had a Mendelian definition of a gene. We had Griffith's definition of a gene. We have a central dogma definition of a gene, right? And we have now a brand new bioinformatic definition of a gene, which has to do not with proteins, not with phenotype, not why you look like your parents in the sense of external appearance, but rather how many times this combination of DNA sequences occur in a genome. So a brand new definition of a gene based on the molecular biology of the relationship of DNA and RNA and protein. So really good way to look at it. The next step, of course, is how do we go for translation from our little RNA sequence to protein? This is the same slide that I use in the lecture materials, and I really do like this slide because it confers the complexity of transcription and translation, the importance and role of ribosomes and our ribosomal proteins binding to the messenger RNA and zipping along. You have multiple ribosomes using the same piece of message, very common, to create our peptide sequence. And this is a really nice graph. I hope that you go back to it and focus on some of the details, but I'm just using it here to remind you. Of course, nascent is another great word. Nascent meaning newly born or brand new, okay? Very often used in a lot of different contexts in biochemistry. So nascent, a brand new, newly born peptide, okay? So we've got the nascent peptide. But of course, this process can be viewed from a molecular biology point of view, which is the purpose of this graphic or cartoon, or it can be used from an information science bioinformatic point of view. And in order to use and to understand the information, we need to recognize that there are 20 amino acids, another a classic picture also used in the lecture materials of all the different amino acids. I want to emphasize, I love this graphic as well. There are a couple of really good ones that we've found over the years. This shows very, very nicely, of course, glycine, you may know, as the simplest of the amino acids. Note our amino group, note our organic acid or carboxyl group, and note our side group or R chain right there. And of course, glycine is the simplest of the amino groups, amino acids, amino acid, and that's it. It just connected by that central carbon. So that's kind of the classic simplest of all amino acids. And you can see how complex some of them get. We've got some amino acids that have ring structures attached to them. Some of them have branched side chains. Here's a weird ring structure. Histidine is a really odd looking amino acid. We've got a variety of terminal groups, everything from hydroxyls to oxygen moieties. Here's our buddy cysteine. If you remember from the lectures, this sulfur group is extremely important because this then binds, I'm not going to draw them all. Okay, that's a COOH. There's the other part of the amino acid. And we get a bond between the two cysteines from two facing amino acids. These cause the disulfide, two sulfurs, disulfide bonds that help to create the tertiary structure of individual proteins. So just kind of overviewing some of the things we talked about in the lecture slides. And of course in the textbook. So really these are very, very nice drawings to remind you of the structure of the amino acids. When you get to biochemistry, I do not know, I do not recall offhand if biochemistry here, they make you memorize the R groups.
Yeah, they do.
Oh, good. I'm glad to hear that. I always have an ongoing chat. We think about both the names of the individual amino acids, which you should be familiar with, even if it's only from reading the ingredient panel on food you eat. But there is also, every amino acid has a name. There is a 3-letter code for each amino acid. These are in parentheses in this graph. So the 3-letter code is very, very simple. Why? Because it's just the first 3 letters of, in most cases, 3 letters of the amino acid. And finally, the single letter code. I often get asked historically, why a single letter code? And the answer is, if you're writing protein sequences, if you're writing out or typing out the primary sequence of amino acids in a particular peptide or protein, it's a lot less typing and a lot shorter for publication purposes if you write in a single letter code than if you use 3 letters, than if you use the entire name of the individual in the individual amino acid. I suggest that you just get used to using the single letter code. At least the last time I chatted with the biochemistry folks, they were not making you memorize the single letter code. Is that still true?
Yeah.
Well, I think you ought to learn the single letter code. So get used to using it. It's really not that difficult. Most of them make sense. There are only a handful of them that are confusing. There are only a handful. The one that actually there are 2 that always mess me up. Tryptophan up here because it's a W, crazy, not the easiest one to remember. And let's see, where in the world? I'm blocking here. Some of them are pretty easy. Phenylalanine, even though its pH is an F, that's pretty easy. But phenylalanine is always a pain. Glutamine is always a pain because it's Q, but mostly I forget tryptophan because it's a W. But you get used to it. The more you use them, the more you'll memorize them. So how do we go from that messenger RNA all the way over to thinking about our amino acid code, our amino acid structure of the protein? And of course, what we have is a translation table. This is our codon usage table. These are classic across biochemistry, molecular biology, genetics, and they are set up understanding that the translation between messenger RNA or DNA and amino acids are based on our triplet codons. And so we have the first letter in the first column. So if we have a triplet codon of U, the second letter is A. So if we were looking for example, UAC, we would find the U, go to the letter A, and then find the letter C. And we would see that this particular UAC triplet codon codes for tyrosine. And of course, tyrosine is a single letter Y. So tyrosine is coded for by UAC. in the previous slide, we noticed a really important codon. And if you recall, the start codon was AUG. So we look for AUG, and notice AUG is in fact the start codon. It is always the start codon in every single species. Every single species. It is also the code for the amino acid methionine, which, by the way, has the single letter code of M for methionine, and it is always methionine. So we've got the start codon is AUG, which always codes for methionine, which means that every single protein that comes off of the ribosome, so every nascent protein, has methionine at which end? Remember, the messenger RNA goes 5' to 3'. If we get AUG as our start codon, this translates to the amino acid methionine, and it is always in the amino to carboxy direction. It is always in the amino carboxy direction. So not only do our DNA strands have directionality, but so do our amino acids. Now, give me a sec here. I'm going to create a white screen here. There we go. I just want to remind you of the structure of our amino acids. I'll put that up there. So here are two different amino acids. Each one is just R1 and R2 to remind you that they have a different side group. And the peptide bond is formed by removing water from, so called a condensation reaction, of course, removing water from the carboxy terminus of one amino acid and the amino terminus of the other, creating, of course, the peptide bond. As I noted in the lecture materials, one of the strongest covalent bonds that we know of. But this means as we string together our amino acids, one end has an amino group, the other has a carboxy terminus. So our 5' to 3' messenger RNA results in a peptide that goes from the amino terminus to the carboxy terminus. Okay, so directionality is encoded by our DNA sequence. So it's always going DNA to RNA. So even though DNA is, the template strand is 3' to 5', messenger RNA is 5' to 3', and the 5' to 3' results in an amino to a carboxy terminus within our peptide or polypeptide structure. So really classic. These, if you want to see, there's some great ways. I don't know. This was one that look kind of cute. Not my favorite codon usage table. There are some printouts that are in Blackboard that you can make your own. Usually on the exam, I'll include a codon usage table so you have one to use, so you can do some of these translations yourself. So what I would like to do is take the opportunity to look at, I don't know what, there we go, at some conversions. These are kind of classic. Okay, so if we were to convert these messenger RNA segments into polypeptide equivalents, again, in order to do this, you're going to need to use the codon usage table. I highly suggest that you take these. They're really not the kind of thing we can do in our space together here, but these codes are in the problem sets in our Blackboard folder. If you're watching this video, you can always do this on your own. Just go back to the video, come up here, pause it. I will tell you that I always give the answers down here so you can see them. One thing that I failed to note, sorry about that, on our codon usage table is the fact that if there is a start codon, sorry about that, I got excited with the peptide bond thing. Notice that there are a handful of what we call nonsense codons. They do not code for an amino acid. What happens is if you hit one of those, these effectively act as stop codons. Once RNA polymerase hits one of these codons, the most common in eukaryotic cells is UAG. UAA and UGA generally occur in prokaryotes, sometimes in some simple eukaryotes, but certainly in vertebrates, the most common is UAG. So these tend to be rather species specific, but there are 3 of them. So when you are translating these into, note here, if we were to translate this mRNA segment, we've got lysine, and notice what happens in here. 1, 2, 3, there's lysine, ACC. Very often I like to just split these up into triplets, and notice right here, UAG. So it means that's a nonsense codon. The chain terminates, and that particular bit of messenger RNA only results in lysine and threonine. There are a few tricks in here because, like in this one, notice the directionality, 3' to 5', 3' to 5'. And all of these, by the way, are in amino to carboxy direction. Another standard or convention in biochemistry, genetics, and molecular biology is the direction that we write proteins and amino acid sequences of those proteins. So here's a great question, and this is something that an example also located in the problem sets in Blackboard and worth chatting about here. This would be a typical problem that you'll see on the exam. Notice that we've got our promoter or transcription start sites indicated in yellow. Here is a classic sequence of what is one of the first promoters identified. As you recall from reading the text, there are several. The most common, of course, is what they call the Tata box Why? Because it's TATA. Okay, pretty easy. So the Tata box moves is the transcription start site. Here, of course, where the arrow is where the RNA polymerase sits down and begins the process of translation. So now take a peek at this sequence. Where does the actual peptide begin? Where does the messenger RNA? Okay, and notice the question says, which strand of DNA, the top or bottom, is the template? So which is the template?
The top one.
The top one is the top one templated or coding?
That's
Remember,
coding. The bottom is your template.
right? And why?
It's your oh, because it's the 3' to 5' end.
And
Right.
what uses the 3' to 5' end? Right? You're exactly right. So you have to remember that the 3' to 5' is the template because it is the direction in which the RNA polymerase works to create the code. It's just one of those things you get used to. So if we were to look at this sequence, we know that RNA polymerase starts here in the direction of the arrow. Where does the message actually begin? So what are we looking for? If we got a transcription start site, what are we looking for next in the sequence?
ATG.
Because that is
The start code.
Absolutely, right on. So we just kind of go along. You just start looking. When do we find an ATG? Okay, I tend to scan these things and look for an A, right? Because we want to be in order. There's an AGC. That's not it. GCCA. Ah, look. ATG. So there is our start codon, which means that if I were going to write, notice question B is what's the sequence of messenger RNA? All right. Well, you'd start writing. I just write it from the coding sequence here. I have a habit of marking off my triplets. I mark off my triplets. And then again, this is just my habit. You can do it however you want. My habit is to take the coding strand because I know the coding strand will match the messenger RNA. So I know it's A. Remember, we've got to replace the T's, just like I forgot last time, right? CAU, TUT. Is that right?
No.
No, what should it be?
UAU.
Ah, very good. UAU. Right, I forgot. I got confused. I started to convert and got all confused. No, just write it out like this and remember to replace the T's with, remember to replace all of the T's with U's. So we end up with UAU, CUA, GAU, AGU, AGG. That's an A. Okay. CUC, UGA, GAA. Okay. Now we can write along these And you may have noticed by now, if we go back and look at the nonsense or the stop codons, here is one of them. Remember, our two of the most common stop codons are UGA in bacteria and UAG in eukaryotes. So here's our stop codon. So if we've got a start and a stop, this triplet doesn't matter because this is where the RNA polymerase will stop. So we go start to stop, and there is the sequence of the messenger RNA. All right. The last question, of course, is what's the sequence of protein produced from the message? Well, the answer is you have to go back to our codon usage table. So give me 1 second. And so we know that AUG is methionine, right? We know AUG is methionine. What is CAU? And I'm just going to go backwards here for a moment. So if we want to know what CAU is, can anybody see it pretty quickly? C, A. So here all right, this column is all the CAs. CAU is histidine. Oops. So if CAU is histidine, the next one is H. UAU, that's the one I got wrong. UAU is tyrosine. So we know tyrosine is Y. And I'm just going to say right to the end, we know UGA is the stop. So the last amino acid is CUC. Oh, how handy. CUC right there is leucine. And of course, leucine is L. So the sequence of the protein produced from this little stretch would be methionine, histidine, tyrosine, the handful of these amino acids. It ends in leucine. And of course, here was the stop codon. We do not need to note or annotate the stop. We just need to annotate the amino acids. Notice, classic question, label the amino and the carboxy terminus always in the same direction as the 5' to 3'. So we have our amino and carboxy terminus here. So this is just classic genetics, classic molecular biology, and really the culmination of our section, this brief little interlude on molecular biology of the gene or the biochemistry of the gene. There are a number of questions and practice problems through the text. All of these slides and problems are located in the problem set folder on Blackboard, so you could get a hold of these if you don't want to come back to this presentation itself, which I will record and of course put in the usual folder in Blackboard. So, seems like this is as good a place as any. Any questions? Just a quick reminder, if you have not started your blog posts, there have been several really, really good ones. Again, I do not comment on the blogs themselves because I don't want to disturb conversation, but there's been some really, really good ones. So I highly recommend you read, not only write yours, but read them. Remember, all the blog posts plus the comments are equivalent of an exam grade. So there are 100 points just waiting for you for free. I gotta say, and I don't think it's anyone here, but there are a couple of people who have not done any. And I know that while there's time left, time is running out very quickly on us. So you really want to make sure and get those done. I hate to leave points on the table. But if you decide not to do them, thank you, because it stops the dean from yelling at me for having grade inflation be rampant at Stockton University. So, any questions, answers? Well, in that case, it's Friday. Go have a good weekend. You guys take
Thank you,
care.
everyone. Thank you. Have a great day.
All right, we'll see you
Take
guys.
care.
Bye-bye.